everyone. So, other than Dirk, as he said, I'm a clinician, and I work at the Center for Rare Developmental Disorders here in Munich. And before we get started, I have two conflicts of interest to declare. I was part of the Red-Grin Phase 1b/2 clinical study sponsored by Green Therapeutics, and I'm still part of the Rett Syndrome Advisory Board. Our goal at the Center for Rare Developmental Disorders is to offer a syndrome-specific clinical counseling. We are all pediatricians, we are not geneticists, and then interdisciplinary long-term patient care for children with diverse, rare genetic developmental disorders. And since genetic developmental disorders are very diverse from their genetic background, through the years we had quite a few patients with very different genetics. This is our gene list from March this year, with monogenic disorders as well as copy number variations. And from this work, a few specialty clinics arose, and one of those is going to be, since this year, starting this year, a specialty clinic for PURA syndrome here in Munich. A specialty of our way of working is, and this is because of this diversity of the genetic background, that when a patient is registered for an appointment, that it doesn't matter if it's the first appointment or a follow-up appointment. First, we do a structured database search. So we have biologists who go through a few databases in a structured way. They look not only for the phenotype spectrum, the current recommendations for clinical care, the support groups and gene function, but also, and this is the topic today, the potential targeted therapy approaches. These are the about 15 databases they go through for every single gene and genetic disorder. So we talked a little bit today about targeted therapies, and also yesterday in the scientific meeting. So what are targeted therapies at all, as opposed to symptomatic therapy? So targeted therapies specifically target the intra or intercellular pathomechanism altered by the genetic disorder itself, and therefore they stand in contrast to the symptomatic therapies, where I just treat a symptom. We have different types of targeted therapies. We have gene therapies, we have novel drug developments, we have drug repurposing approaches, and we have this big group of diet therapies. From the efficacy, we differ curative therapies and non-curative therapies, and at the current state for genetic developmental disorder is that most therapeutic approaches are of non-curative nature. From the status of approval and therefore availability in the daily clinical routine, we differ approved therapies. They have to be approved for the indication, for the age, for the route of administration in the country where you work at. Prior to an approval, when an approval seems to be very likely, we have compassionate use programs. We have clinical studies, phase 1 through 3, for an approval goal, and we have scientific clinical studies, and we have off-label treatments. They are usually drug repurposing or diet therapies. And since we were doing this way of working for quite a few years, last year we conducted a single-center retrospective observational study with the two main questions. How often were we actually able to identify a targeted therapeutic approach for the genes with which patients were coming to our center, and how often our patients got a targeted therapy initiated? And of course, we wanted to see the efficacy, the underlying evidence, and what was the availability of these targeted therapies. And so we included all patients we have seen ambulatory from June 1st, 2021 and December 31st, 2024, with a monogenic neurodevelopmental disorder. And therefore, we excluded or CMVs or copy number variations without a candidate gene, also excluded disorders of methylation and patients with disorder of the physical development only without a cognitive impairment. And so we landed with 222 genes and 348 patients in pediatric ages, and as it is with rare disorders, for most of the genes we had only 1 or 2 patients, with a few exceptions. And to the results, we were very much surprised ourselves that for almost 25% of the genetic disorders, we were able to identify a targeted therapy, regardless of if it was effective, if it made sense, but it was published in a peer-reviewed journal, or there was a clinical study. And in 15% of the patients we have seen was a targeted therapy initiated, not always through our center, sometimes from other centers. From the status of approval and therefore availability, we have seen that approved therapies and therapies within an interventional clinical trial were much rarer, and especially they were rarely initiated. And honestly, these 1.1% for the interventional clinical trials, those were 4 patients. 3 of those were in the red-green study, where we were one of the study sites in Germany. So the vast majority was drug repurposing, also from the identified and as well as from the initiated therapies. From the underlying evidence, we have seen that most targeted therapy approaches were based on a case series. Those were the therapeutic approaches we initiated in our center. So 40% was based on case series, 28% was based on clinical trials, 24% was based on pathomechanism. That is a sort of expert opinion. So if there is a publication, if there is an expert who has a thorough understanding of the intracellular mechanism, and there is an idea of what kind of medication could help. And in 8%, in 2 cases, they were based on studies in animal models. And so to the effects of the in-center initiated therapies, there were 42 patients, and these in-center initiated therapies were all drug repurposing approaches. Drug repurposing is done within the regular patient care. So first, for this study, we had to define what do we call a positive effect at all. And it is not so easy because the patients are different. You've seen there are quite a few genes and quite a few therapeutic approaches we have done, and therefore how we measure it has to be also different based on the approach and based on the patients, the age of the patients. So we defined a positive effect as either objective data, such as a developmental test or a gait analysis, showed a better than expected development, or if there was a positive decision for a long-term therapy together with the parents' doctors after a therapy phase and a planned withdrawal phase. So that was very important. There was a planned withdrawal, and after that we together decided that the effects were good enough to go for another 2 years with the therapy. And using these criteria, 34 of these 42 therapeutic approaches or therapies offered were evaluable, and of these 34, 18 showed positive effects using this definition. Well, 18 from 34, that's just a little bit over 50%, so you might say, well, that's not so great, not everybody benefited from the therapy. But if you compare that with trofinetide, trofinetide is the first novel drug development for genetic developmental disorder, for Rett syndrome. It was approved through the FDA in March 2023 in the US, and it's currently in the compassionate care use program here in Germany. And these are data from the phase 3 study. They use CGI, that's the Clinical Global Impression Scale. And the CGI scale showed for patients getting trofinetide debut much improved or minimally improved in 37% against 15% in the placebo group. And still the medication got approved through the FDA. And these are the approaches, the therapies we offered and initiated in our patients. And not on this list is this salbutamol therapy for one patient with PURA syndrome we initiated last year. But unfortunately, since we are not done yet, because the patient is coming back in September for the end of the therapy phase, I would love to present it next year. We have more data then. And then we wanted to see what kind of symptoms can we expect to get better through drug repurposing approaches. Obviously, a drug repurposing approach is never curative, right? But what kind of symptoms can we say that, well, that might get better? So we have seen that the most common symptoms showing improvement were speech, followed by concentration, cooperation, motor development, and sleep. And I brought you one use case to show you how the idea of a drug repurposing approach is actually evolving and getting to the patients from the bench to the bedside. And this use case is from Pitt-Hopkins syndrome, because this is also a specialty clinic and a drug repurposing approach we apply, and the case of nicardipine for patients with Pitt-Hopkins syndrome. The Pitt-Hopkins syndrome is also a neurodevelopmental disorder based on the haploinsufficiency of the TCF4 gene. The symptom spectrum is the first three is quite similar to PURA, moderate to severe intellectual disability, with a prominent speech delay. Most patients are also nonverbal. Behavioral differences with symptoms of autism, sleep disturbance, stereotypic hand movements, and epilepsy. But they also show a very marked autonomic nervous system dysfunction, episodic hyperventilation with severe breath-holding spells, and a severe myopia as well. And before we get further, in this case, I wanted to show you how we think when we think about targeted therapies. So TCF4 gene and also the TCF4 protein works in the cell nucleus. I have no medication to get into the nucleus. In the nucleus, it's only a gene therapy that gets in and can work in that area of the body. Medications can work on the cell membrane, the synaptic level, that's the second here. And therefore, what is important to find out what, for these drug repurposing approaches and also for novel casual medications, is what are the effects on the cell membrane synapse level where a medication can act. And obviously we have the brain dysfunction as a global symptom. We also have approaches for the brain dysfunction as a total, but those are like a deep brain stimulation, but those are actually currently more used for movement disorders. So getting back, here the first step was to identify a potential, there's something missing, a potential therapeutic target, and that was through a mass spectrometry molecular profiling of the translitome, and there was a sodium channel. And here you see in red that this sodium channel was much more active as a total. There was an overexpression of this sodium channel compared to the other channels they had studied. And then Sean Akins did the studies and the drugs screening studies and identified nicardipine and nilvadipine as the most potent inhibitors of this total sum overaction of the sodium gated voltage gated sodium channel 1.8. And then they went forward, and that was actually the publication we have found through our database search, and they did studies in knockout mice. And that was the study where we said that these are such impressive results that we got to think about to offer it to our patients. And I walk you through here. The black box is the healthy mice. And they did quite a few studies. I just picked these three. Nesting in mice is a normal social activity. So we want to have that. The healthy mice in black ball does a lot of nesting. Fear conditioning is learning, and also the healthy mice did good learning. It's a good ball. Self-grooming is more like an autistic features, so a healthy mice didn't do a lot of self-grooming. As opposed to TCF plus minus, that the knockout Pitt-Hopkins mice, they did less nesting, less normal social behavior, less learning, less fear conditioning, a lot of self-grooming, a lot of autistic features. And if they treated these two mice types with nicardipine, you see that in gray is the wild type, the healthy mice treated with nicardipine, not much changed. But the very impressive is the yellow or green bug, that is the knockout mouse treated with nicardipine. And actually, nesting is almost back to normal, fear conditioning learning is almost back to normal, self-grooming is also almost back to normal. Now, they used 3 milligram per kg, that's an extremely high dose. We never got there in patients, so that is the kind of thing what is, the difference between bench and bedside, what is possible and what not. And this is our first patient we have treated, a young girl. And here you see the blue box, the different developmental areas, and we talking of different developmental ages, gross motor scale, fine motor scale, speech, and perception. And perception is the one scale which has the best association with cognition later. And you see that at the age of 32 months, when we first met her, she was in all developmental areas under 8 months of age. So it's a severely affected girl. And didn't much change until we started nicardipine. That was at the age of 38 months. And at the end of the nicardipine treatment period, at the age of 45 months, she made a good effort. Good progress in all developmental areas, especially also in perception, because that for us developmental pediatricians, making progress in perception is a very big word, because this is what we want to have. So we published this case, and we were very much surprised about the echo, because we just published it last year. And I got a lot of emails, and now we treated 4 more patients, or started treating, and there's 1 patient in Brazil, 1 in Japan, 1 in Hungary who got treated, and just yesterday I got another email from Switzerland. And our next step is to apply for the orphan drug designation for nicardipine, and we are very happy that Dr. Aristiziba Prada, she's also a Pitt Hopkins mother and a biologist, she is helping us on this way from the PTHS Society. And for the outlook, so with our regular clinical work, what we wanted to do is to get science to our patients. So what is out there, what is possible to understand, and if it makes sense, get to the patients and initiate targeted therapies. What we also wished for is to have scientists to do science for our patients. So the other way around, so therefore we just applied for the ethic approval for our project Tutti. The name is coming from the world of music, where everybody plays together. And the project is called Together to Understand and Treat Genetic ID. And here the goal is to identify new pathomechanisms and new drug repurposing approaches. And so the patient recruitment and clinical characterization is first in our center. And we have quite a few scientific cooperation partners here in Munich, University of Bonn, University of Heidelberg. And we also have a cooperation with the German mouse clinic, so in case there is a targeted therapy identified, and could be also some study done in mouse model. And my acknowledgements to our team and to our cooperation partners. Thank you for the invitation and to be here, and thank you for your attention.